From Microbe TV, this is Immune, episode number 31, recorded on May 6th, 2020. I'm Vincent Racaniello, and you're listening to the podcast about the body's defenders against disease. Joining me today from Ithaca, New York, Cindy Leifer.
Hello again.
How have you been? Well?
I have been well, healthy, working from home, not leaving the house much. But it's very interesting. I think I can get almost everything done that I need to get done here. I just am excited to get people back in the lab. We got some pilot COVID funding, so we're going to look at some interesting things coming up.
Good. From Durham, North Carolina, Steph Langle.
Hey there. Good to be back. Part 3 of our COVID immunology conversation.
How many parts is it going to be? Like 100?
Well, yeah, I know. We really could just keep this going for a long time. I don't know when people are going to want to hear about different things, but for now, there's quite a bit to discuss.
I don't know about you, but putting it in context that, I'm teaching an advanced immunology class right now and sort of using this as a method to, as a way to teach the information. The students are really appreciating that because they're getting some information about this current thing that's going on, but then putting that in the context of their learning. I think it's a unique and really exciting opportunity.
For sure.
Yes, I agree. And from Madison, New Jersey, Brienne Barker.
Hi, it's great to be back with you guys. I'm excited to talk more about the immune responses to coronavirus.
What's that? Never. If you like what we do on Immune and all the other podcasts, consider supporting us. Microbe.tv/contribute. Let's do some email first before we talk about immunology.
That's a great idea. I think the listeners have been writing in like crazy, and we've been just discussing normal stuff.
So Lucian writes, there was some discussion on Immune 30 about the feasibility of driving across the U.S. in about a day. This is a known time trial called the Cannonball Run and has been done. As you all pointed out, this requires driving for long hours in great excess of speed limits, and so is both very illegal and very, very dangerous. Nonetheless, it's been done. Here's a story of one pair of racers from Wired magazine in 2007. Provides a link. As always, thank you for all your insights and humor during these very strange times. Yes, indeed. I guess think, the humor with the Cannonball Run.
Yeah, I don't know that anybody's going to beat the record that these people set while the pause was in effect without the cops out there and the cars out there. I don't know if it's going to be able to be done. I think I looked at that article she put up, and I think they did it in 37 hours, and I think I remember saying it was 27. So they should. I don't know if that's going to be doable again. Do you?
Right.
Was it before you were born? But do you remember Cannonball Run, the movie?
I do.
It was a comedy movie, but it was probably, I don't know, Stefan.
Yeah, that I'm not
too young.
I'm not.
I am not familiar with it.
Oh no, that's terrible.
That's
now
very
I'm checking. Oh, I was born.
You were born. Oh, yay, good.
What year?
I don't feel quite so old.
It was in 1981.
I was pretty young. I was pretty young.
I was.
I do remember it.
I was someone's memory for 7 years, and then I was a thought 7 years later than I was here.
Yeah.
Oh dear.
Oh boy. But that's interesting. Yeah, I mean, now that people are at home, I mean, I wonder people could. Not that I'm promoting doing illegal and very, very dangerous activity. But if you did it now, you might be able to beat that record.
Just don't go through some of the states they opened already, right?
Well, that's true. Wear a mask. So yeah, we do have a couple more emails. I'm jumping down that we could address before we kind of get rolling. It's easy to get rolling and then we don't get a chance to get to those emails.
Yeah, that's what we've been doing.
So
Sophie's first.
Is Sophie first or Jesse?
Yeah.
Oh, you know what? Jesse we'll talk about in the context of that conversation.
Sounds good.
So if you want to read Sophie's, that's fine.
Oh, sure. Okay. Sophie writes, Dear Immune, I am a student in BI 115 virology at Caltech. Hopefully I said that right. And we recently discussed the bat immune system. One thing I found quite interesting was that bats have to suppress their immune response due to the inflammation caused by their high metabolic activity. could people with higher metabolism, which I believe is associated with exercise, also have increased inflammation that could lead them to have a suppressed immune system that would allow them to tolerate the virus in a similar manner to bats? So I think this is really interesting. And I think that one of the things we have to think about is the metabolic activity issue. So there's metabolism that you think about where skinny people have a quote unquote high metabolism, or exercisers, right? But the metabolic activity that we're talking about here, I tend to think of it a little bit differently, is how we're using glucose within cells. And so there's an interesting thing in connection between inflammation and glycolysis, and that might be different, but I'm not sure that the runners are going to have a suppressed immune system. I think they'd actually have a more active immune system. I don't know what you guys think about this, but I know that. Go ahead.
I think so. I think the changes in bats are quite a bit more dramatic than what we see in people, because they're associated with flight, as bats being the only flying mammal. And so I think that that's longer term than we see in people, and much more dramatic. And with bats, this has led to sort of some long-term evolution of their immune system, in terms of changes in immune genes, that I don't think we would see happening long-term in humans who were running.
Right. And some of the things that the bats have are like changes in their innate immune recognition signaling mechanisms. Right. Because I found an interesting paper that came out last year, I think.
That's
on
such
Sting.
a great paper.
I love it. I think we should do it on our immune podcast. But yeah, so they have defects in the signaling responses to viruses, which, they seem to be more resistant, but yet have a lower immune response, which is kind of a fascinating thing.
It is.
And I think there's just so much more we need to know about bat immunology. Right?
Yeah,
I know.
definitely.
I have learned that they are actually missing all of the genes in the family that I study in my lab.
Oh, wow.
Right.
And so I often flirt with the idea of starting to do some experiments on that.
Oh, you should.
Unfortunately, I don't think NIH is going to fund them.
Yeah.
I know.
Well, you can use bat cells.
Yeah, you
right?
can use. Are there bat cell lines? Probably.
There
Sure.
are, definitely.
Okay.
There are.
Yeah, we have one. We just got it from Ian Lipkin.
Oh, good.
Oh, cool.
Yeah, I've thought about getting some because, the entire gene family that I study.
And which family is that?
All of the PIHIN proteins, so IFI16 and AIM2, all of those PRRs.
Yep.
Oh, nice.
DNA sensors.
The problem with studying bats, and I talked about this with Linfa Wang, the TWIV we did a couple of years ago. So, they have bat colonies, right?
Yep.
But they don't fly around. They're in
Right.
cages. So they're always resting. And so, yes, it's an issue because you cannot get them to the point where they're, flying and they're like 105 degrees Fahrenheit temperature. It really cranks up. So it's hard to study.
Yeah, unless you're doing field work and capturing bats, of course, then that's going to be a stress-induced response because they're not going to want to be captured.
Yeah.
Right.
Yeah.
They do have large cages in which they house bats some certain places for research.
Yeah, but these labs can't afford so many of those, right?
Right.
'Cause-
I imagine it's very expensive work.
So I was supposed to go to a bat meeting this summer in Fort Collins, and of course it was canceled. But they were going to give me a tour of the bat colony, and that would have been cool. Then I could speak firsthand.
So cool.
I can imagine them putting the bat on a long leash and letting it fly around.
Did you ever see those things for Halloween where you hold it and the thing flies around in a circle on a string? That's what it reminded me of.
But we could definitely do this paper. We could also try to get, Linfa Wang is probably pretty hard to get a hold of right now with COVID, but we could try, a postdoc or another.
Well, there's a guy in Fort Collins who invited me. He's a bat guy. I can't remember his name, but he would
That
do
would be
it.
great.
Yeah.
I could do it sooner than Linfa.
Yeah, sure.
And the time difference with Linfa is difficult.
Yeah.
For sure. Touching back to, I think we had discussed exercise-induced inflammation before, and we had made the point where it really is a difference between an acute versus a long-term effect of inflammatory proteins on your body. And also, thinking about excess adipose tissue, that also comes with its own inflammatory milieu. So
that's right.
I think it would be very challenging, and probably why exercise immunology is really, that's a difficult field to try to parse through. So, but great email, because I think it's going to lead to an episode or two.
Yeah. Thanks.
Yeah.
So why don't you take that next one, Steph?
Yeah, sure. Whitney writes, hello, my name is Whitney Sloniker, a BI 115 student from Caltech. Seems like they're in the same class. I had a few questions based on your Immune 29 podcast. You mentioned that envelope viruses tend to be more unstable than other types of viruses, but coronaviruses seem to be stronger in that they enter the digestive system. What mechanism would you theorize is responsible? Do you think the differences regarding how IL-2 reacts in these different regions plays a role in ensuring we see stronger symptoms in the lung versus the gut? Are there different levels of SARS-CoV? She put 19, but I get it. I think she's saying like COVID-19 and SARS-CoV-2.
Let's hope we're not at 19.
No, we're not at 19. In both regions, do the structure of the virus and does that play a role in the effects we see if present? So yeah, I mean, coronaviruses, they can enter the gut even though they are enveloped viruses. So clearly they have enough stability that they're able to last through the pH of the digestive system. And then after that, it's really all to do with the receptor, which for most of these is an enzyme, as to whether they're going to bind and cause disease. You know, comparing coronaviruses as a more stable virus than others, I don't know. I don't know if I want to speculate in that way, other than just to say for coronaviruses, it seems fine. And then what mechanism would you theorize is responsible? I would imagine if we were to break down so that the structural proteins, the membrane and the envelope, I'm going to assume those play a role in keeping that virion intact. And if we compare between families, maybe that's where we would see differences. So you could kind of focus on those structural proteins. And differences in how IL-2 reacts in the lung versus the gut. I don't know about IL-2. I mean, maybe she meant IL-6 because that was a driver of pathogenesis.
Yeah, I'm not sure.
Not sure about IL-2, but
we know IL-2 is important for T cell proliferation.
Yeah, right. So IL-2 would definitely both play a role in those two organs. Let's say it was IL-6 that she meant. I think that the IL-6 is driven by macrophage infiltration, which can have negative effects both in the gut and in the lung. So you can think about immune infiltrates coming in during inflammatory Crohn's disease or IBD, inflammatory bowel disease. So they definitely play a role, just in a different way. Her last question, different levels of SARS-CoV-2 in both regions. So that's interesting because we know the receptor ACE2 is expressed in both the lung and in the gut. And there are high viral titers in the gut. I don't know if we can say with much confidence the transmissibility of that quite yet.
Yeah.
Because a lot of these studies are not using our favorite plaque assay to determine if they're infectious. But it may play a role. I think we're going to learn more as papers come out.
What do you think about the studies that are looking at like in hospitals are saying that the highest levels of virus that they can detect, the viral genome that they can detect by PCR are in the bathrooms. Do you think it could literally just only be non-transmissible form that they're detecting just because it's massive amounts that people are pooping out?
I think that could be viral remnants.
Yeah.
Yeah, it's Christian Drosten said they can't find infectious virus in stool
That's
at all.
Which
so
is
it
just
pieces,
Go ahead.
Well, and I know coronaviruses in general need greater genomic content to form an infectious plaque. Yeah. I would have to add more than another RNA virus. So, and then if those are all degraded, I guess that could contribute to this high load. But right, it's still wondering why is there so much there, even if it is degraded?
Yeah.
Well, it's just becoming a general phenomenon with RNA viruses that you get a lot of pieces of genomes because we can detect them independent of a plaque assay or infectivity assay. And Diane Griffin thinks that this happens with basically every RNA virus infection. Sure. There's a tail after the infectivity is gone. You still have this tail of pieces of RNA that go on for a long time.
Sure.
Maybe a month.
How much do you think coughing and swallowing of virus might lead to detectability in feces, for example.
No, that too. Sure. You can get flu in the feces, flu RNA, because people swallow it. Yeah.
Yep.
You swallow your mucus. That's how your ciliary elevator brings the mucus up and you swallow it.
Right.
And then, so yes, that's what I think when people find, we got flu in the feces, it's just swallowed virus. But in this case, it seems to be reproducing in the gut.
Well, that's the thing. I was going to say, to get this high of titers is what we're seeing in preprints and papers. It would have to undergo replication at least one cycle, but maybe it doesn't have an additional enzyme. I mean, there's other enzymes, cathepsins, that TR and PR.
Now, when you say titers, you mean RNA copies.
I'm sorry,
right?
RNA copies. Yeah. Not infection.
Because it's
you can't get sloppy just because we're on immune, right? You got to check
this
there. Make
paper
sure using the right viral
on
terms.
saliva. Did you see this paper on saliva detection a couple of weeks ago from Yale? They call it virus titer, and I wrote to Kiko. I said, "No, you can't say that. It's absolutely wrong." Oh, no. I don't want to yell at people, but please use your terms properly because the public is really watching now.
Right.
Oh, yes. And they're all.
But the thing that's funny is that it's not even genome copies, right? Because you're just amplifying a small piece. So
you don't know if you have every part. I
guess it should be amplisomes per mil or something.
Okay.
Right.
It's very what's the word? Very.
Nuanced?
I can't think of the word. No, it's a
precise, specialized.
No, not precise. It's
technical.
It's unwieldy. It's an unwieldy term to say amplicons per mil.
Yeah.
I can no longer think.
We've reached the point where it's just like it's too many Zoom meetings, too many papers.
By the way, these two from Caltech. So we learned over on TWIV that they're part of a virology course, and the instructor assigned them to send in emails to podcasts, TWIV and Immune. So that's where I forgot who the instructor's name is. Do you remember, Bria? Yeah, Bjorkman. Yeah.
Oh,
Bjorkman. Yes.
Oh, great.
Yes. So she's having her students, and in fact, we don't even have to answer them as long as they send them in.
Well, we'll make sure to answer them. Yes. Do we feel like we answered Whitney's question appropriately?
I think so. Okay. I think you did a great job.
So,
anything that you wanted to add? So, Vincent?
No, we're good.
Okay.
Maybe Brianne can take the next one.
Can take the next one.
Okay. Matt writes, hello. Brianne Barker recently noted that there was a third type of immune system, which is distinct from the innate and adaptive immune systems. I recall it had something to do with memory cells. I would love to hear you discuss this on a future episode of This Week in Immunology. Keep up the great work, Matt. So I think that this is a reference to a comment I made on TWIV about trained immunity, which is not actually distinct from innate and adaptive immune systems. I wish I could take credit for coming up with a third type of immune system. That would be pretty great.
Don't forget about us when you win the Nobel Prize for your
I definitely won't. I definitely won't. So I think this is when I was discussing trained immunity, which is this kind of new area of study that people are looking at, which is kind of an innate memory response. Yeah. So it's not really distinct from the innate and adaptive system. It is part of the innate immune system. It involves innate immune cells, but they are acting in a way that we normally associate with the adaptive immune system, in that they are acting differently upon later exposure or after exposure to pathogens. So a newer area of immunology, something that I've been reading up on a bit, since most of it was really understood after I left graduate school, making me feel old. But
don't even.
But I'm pretty sure that is what Matt is asking about. So we can talk about trained immunity, but I don't think that it's something distinct. At least if I knew about a third type before, I don't know about it now.
But it was a fundamental shift in our thinking about innate immunity, which I think is just absolutely fascinating.
Oh yeah, it's really fantastic. And there have been a couple of sort of therapies that people have been talking about with COVID-19 that seem to take advantage of trained immunity. I've really enjoyed kind of taking all of this as an opportunity to learn a little bit more about it myself. And it's a total shift, and it's totally fascinating. But does also tie into the fact that we know about lots of different organisms that don't have adaptive immune responses. And they also seem to have these responses where we see changes after infection. And so this may be kind of a very broad type of immunity that's present in many organisms.
Yeah. And it's something, so Brianne brought up a good point that people are looking at this trained immunity as a mechanism to boost people's protection against coronaviruses. And so if you guys are fine with it, we can roll into that conversation. Particularly with two different types of vaccines. So one that we call BCG and the other polio, which Vincent did a recent TWIV about this. So I think 604 is the number if you're interested in learning even more about this concept. But, involving different reprogramming of innate immune cells, so whether that be epigenetic or metabolic, is the concept behind a non-specific vaccine boosting your immune system against another type of virus. And we were going back and forth and conversating about this. And I can appreciate that this would not provide herd immunity because it's not specific. This is not going to be specific against the coronavirus, but there's some evidence out there, and maybe Vincent and Cindy, you guys can talk more about that evidence, but it showing that countries that have the BCG, so this is the, what's the French word for BCG?
Bacille?
Yes, Bacillus calmette-guerin. Yeah. So it's a attenuated strain of Mycobacterium bovis, so it's against tuberculosis. Gosh, it's been around for a long, long time. And
they started using it in, the early 1900s.
Yeah. And it's the only tuberculosis vaccine we have. And it's used in countries that not in the US, but that have high, rates of tuberculosis. So comparing countries that had populations that had gotten this vaccine, they had seen some evidence that maybe it could be protective against other viruses. But does anybody have any, I think Cindy maybe wanted to talk.
I saw another paper that said when they broke it down, so there are certain countries that stopped using it. And if they looked at if they broke down by age group for those who would have had it and would not have had it, because after they stopped using it as a vaccine, they didn't see any correlation with the incidence of disease. So I would take that to mean that looking at these bulk countries, like saying that one country uses BCG and they had lower levels and another country doesn't use BCG and they have higher levels. But when you look at the country that did and you break out the population that had the vaccine or had not had the vaccine, there was no difference. So I think that this is not, I wouldn't say debunked, but I think it's quite questionable whether this is true. Yeah. That being said, if you look at the basic level, what the active or most active component of BCG is from Coley's toxins way back when is DNA. And DNA triggers TLR9. And there have been studies, for example, from the lab in which I did my postdoc, where we found that, just injecting cpgDNA or a TLR9 ligand repeatedly over time and then challenging animals, they were more resistant to infection with a variety of different agents, including bacterial agents and viral agents. And so the idea was, and this was way before we had this idea of trained immunity or any other innate immune memory or anything, was just we had the idea that triggering TLRs on a regular basis sort of kept your immune system like primed to some degree, and so it would make you generally able to respond more quickly and more robustly to any kind of insult, didn't matter what it was. So I'm still not convinced, but I mean, if you looked at people who had recently had that BCG vaccine versus ones who had it years and years and years ago, maybe there's a difference, but I don't know.
Yeah, that's my take as well. I think that there are kind of two sets of studies on this. One, that are sort of epidemiological studies where you're looking at the bulk country data, as Sydney mentioned, and some where people who have recently had a dose of BCG have been examined. And there are some clinical trials going on where healthcare workers are being given BCG. There are some pros and cons to that because of issues with BCG. But the idea of trained immunity, at least from my extensive learning from reading 2 reviews, is that this seems to be a change in innate immune cells that last between 3 months and a year.
Yeah.
And so perhaps people who have recently had BCG might have a sort of improved activity of some of their innate immune cells, particularly monocytes or macrophages, that let them act a little bit better against some of these viral infections. So it's not specific. It probably wouldn't lead to herd immunity, but perhaps it's some way that people at higher risk might be protected for the short term.
There are 8 clinical trials ongoing in 6 different countries to test this.
Right.
All for mostly healthcare workers, but not all of them. So we'll see.
Yeah,
if it has
we'll
any
see.
effect.
And I guess if it's healthcare workers, maybe it would not be people who are, they're at high risk for being in contact with high amount of virus, but maybe not high risk age group wise. And so I do wonder about boosting your innate immune response for a disease that is known to be most severe in people who cannot control their innate immune response.
True.
So, the trade-offs there, I just don't know if we know, what this would do to a person who is already susceptible because they don't have the ability to control their macrophages infiltrating into the lung. So I think it'll be, interesting to find. Now with poliovirus, Vincent, there's 2 different types of vaccines. And so this would have to be the live attenuated version to have effects?
Yeah, that's what Kostya Chumakov said. You don't get this effect with the inactivated polio vaccine. So yes, it's OPV, and he's trying to do a trial. I don't know where that stands now.
Sure,
but
are they doing these
OPV
trials?
Is relatively safe.
Are they doing these trials prospectively? So are they enrolling people and then vaccinating or not, or are they doing these retrospectively where they're looking back at? No,
they're doing prospective trials, and they would be in healthcare workers too. Yeah.
Okay.
And healthcare workers are the real target. You just need a couple of months of protection, right? That would be good.
And with BCG, the concern to reintroducing that vaccine into countries that aren't using it, my understanding is that we sort of use this as a surveillance, right? So
exactly.
TB, multi-drug resistant TB spread is a real concern worldwide, and not being able to do surveillance for individuals who might have TB versus those who don't is a problem. And we use the TB test, the time, pricking your wrist and do the delayed type hypersensitivity reaction to determine if you've been exposed. You'll test positive if you've had the BCG, which creates a whole nother layer of surveillance to do lung, radiographs and things to determine whether you're actually harboring the bacterium or not.
It's interesting that the BCG trials are getting so much traction, and I just looked. There's no OPV trial and clinical trial.
Do you think that's because they want to stop using OPV? I don't know, because the issue of
I think Kostya is just not as forceful as the other people involved.
I think that's part of it, and I think that sort of coincidentally, a lot of the original work on trained immunity has been done with BCG.
Mm-hmm. Yeah, right. Like we understand it better.
Yeah,
but remember, listeners, these trials cost money.
Oh my gosh,
someone has to pay for them, and so if Kostya has an idea, it doesn't just happen. So,
right.
Yeah.
I think these BCG trials are in many different countries. So clearly the countries are behind it. So that helps.
So, big picture. Okay. So let's say that we could get a boost in immunity for 3 to 9 months with the BCG vaccine in healthcare workers. I could see that would be beneficial. I don't foresee this being anything that we could roll out to a large, to society or to the large group, just because I don't think we know enough, and particularly the high risk groups. But if it was focused and it did show it worked, to at least to tide us over, because what we're going to talk about in a little bit is that a vaccine against SARS-CoV-2 is going to take some time, contrary to what you may have heard.
You also might have been quite on the young side, but in 2001, when we had September 11th, and then we had the anthrax threats. Mm-hmm. So that was when I was in this lab, and we were talking about stockpiling CpG DNA. So whether we could, inject ourselves to give a boost of immunity.
Sure. Wow.
Exposure.
You do what you
We never did that.
My lab right now.
We never did that. But the point being, though, that if a primary active component of BCG is DNA, it's somewhat safe, right? Mm-hmm. So it wouldn't have to necessarily be just BCG, which could have its own problems, but you could use DNA.
Yeah, I think that would be much safer than using BCG. I would be concerned about using BCG in whole populations, but something else like DNA could work.
There's some component of it's a live bacterium and it's infectious poliovirus. True. And the inactivated vaccines don't have the same effect.
Agreed.
So it's not, it's not just DNA, but, right?
A little boost is better than nothing.
Well, that brings
that's not a very
This brings the question, if you have influenza now, will it protect you for a couple of months against SARS-CoV-2 infection, right?
I would
infected virus.
Yeah, I would say no. Because
why not?
The reason why
general boosting is the idea.
It's general boosting, except that influenza is infecting the same target cells in the lung and causing tissue damage, which is I don't know that that's going to boost immunity or make you more susceptible.
I would go with, I do the concept of boosting innate immunity, but then I think influenza particularly infecting the same cell type, and then you're going to follow up with this virus, I think it would predispose you potentially.
Yeah, pathology in the same place twice.
Kostya seemed to think that flu mist would have the same effect because that's not damaging cells. I guess maybe that's.
But you know what? We could probably find that out because they did reinstate flu mist this year, didn't they? So there will be a cohort of patients who received flu mist versus the regular flu, and we could look retrospectively to see.
Sure. But we couldn't decipher. Yeah, no, we could do that. You're right. We could.
You could look at people who didn't get a vaccine versus flu mist versus inactivated and see whether there's any difference in attack rate with SARS-CoV-2.
Okay. Well, who wants to write that grant? Ready? Go. Cindy, when your postdoc studies, when you were able to see induced effects of multiple CpG injections, did it work if you just gave it one time? Or was the multiple important?
So I didn't do those studies in the challenge studies, but it's the general consensus was you would inject, you would get a little boost for about 2 weeks, and then it would start to wane. So they tried to do this thing where they were going to boost every 2 weeks. But if you continue that for very long, you end up with some spleen issues because you get some overzealous inflammation and splenomegaly and lymphadenopathy.
Not fun. Well, good. Well, so we feel like we covered BCG polio trained immunity pretty well.
Yes. Yeah. And the fact that there is no third immune response.
No Nobel Prize yet. You got to keep working.
Keep going.
Sad.
So the next thing is something that I've gotten asked about a couple of times, and Vincent recently did a TWEVO about it, if I'm not mistaken. But there's a potential mutation in the spike protein, and a lot of conversation about mutations in coronaviruses, which we've discussed on this podcast too. You really can't say without functional studies whether or not a mutation is weakening the virus or making it more, transmissible. And Vincent, I'll kind of let you talk about this if you want, but it's been something that's been in the news because of this emergence of this particular mutation within more European strains that had seeded into New York and then went back to Europe. So
this is a preprint. Now that's the problem.
That's the problem. Yeah.
And as it is written, it will never be published with using the same language. It's out of Los Alamos, and it's a computational biology analysis. They have a pipeline for looking at changes in the genome that may persist. And they identify this amino acid change in the spike, which they think has been spreading extensively in certain areas. Okay, that's fine. But then they say it's alarming spread. And in the title, it says this mutation, this amino acid change enhances transmissibility, which is where it just falls apart because there's no evidence. Just because a change is persisting doesn't mean that it's doing anything. It's just So there have been a number of discussions about this. What's the guy's name in Washington? Trevor
Bedford.
Yeah.
He agrees that it is probably a founder effect, which means And I told the reporter this yesterday. When you get a virus infection, you're full of marbles. Imagine each marble is an infection. They're a virus, and they're each different colors. You transmit one of those, you transmit a blue marble, and now that person will transmit blue marbles. And so it's just random and has nothing to do with any property. In fact, in the paper, they didn't look at any biological properties at all. So I think it's fine to report this. It's interesting that, a particular change has been sustained, but it is really irresponsible to say it enhances transmission, because all the papers are picking that up. All the news sources are picking that up, and they're scaring people. And I have to say, this is not the first time this has happened. It has happened with Ebola virus. It's happened with Zika virus. People seem to want to watch the virus change and get worse. I don't get it. Anyway, Nels and I talked about this yesterday, but the fact that Trevor agrees that it is a founder effect, which means whatever virus gets into a population, all the others are derived from it, right? And they have similar mutations. And there's no evidence of what we would say positive selection, which would be if this confers some property on the virus that's selectable. And I think that's the other aspect you have to think about. If you want to say a change increases transmissibility, then you need to show me what's the selection for that. Because last time I looked, this virus is pretty transmissible. It's doing a great job.
Yeah, it's doing a very good job. So, we do know, though, there had to be a mutation in order to get it into humans, right?
That's right. A long time ago, maybe last year. Yeah.
Right.
Yeah.
And then there's probably a mutation that allows it to spread from human to human, right?
Yeah.
Because
as Nels said yesterday, all that probably happened long before we saw it.
Right. Yeah.
Because to see it working in people, it's already gone through all of that change.
Right.
And we would love to be able to watch that in action, but we can't.
Yeah. Well, and
we need to know a lot more about all the viruses in all sorts of different organisms to do those things.
Yeah, we should be funding more bat research.
Oh, you mean we shouldn't be cutting it?
I was going to say, I knew I had to trigger something. Yeah.
Go figure.
So I say, okay, yeah, I agree. I mean, I think while there's a hypothesis, and while that could be plausible, we have no evidence right now that would be the case. So people listen to Tweevo to get more info on that, because that would be pretty good conversation. And, talking about vaccines and vaccine development, I think one of the things we all get asked a lot, because gosh, it's just such a part of the conversation in the media, is when are we going to have a vaccine? How long is that going to take? I just think it's irresponsible. And I even think Tony Fauci did say this, although I'm a big Tony Fauci fan. He did say at one point 12 months. And I just think that's irresponsible to say, because 18 months it would be a dream based on what we know of how long it takes to produce vaccines. I think I'm looking at this chart that maybe 10, 11 years was the fastest that we've been able to do. And I don't want to scare people to say it's going to take that long, because there's a lot of investment going into creating a vaccine for basically the entire world. But we just got to be careful. And really what speeding up a vaccine production would mean is maybe skipping out on some safety trials, which I don't know.
Yeah, so I think that there is some acceleration pace of development, right? So, computers and technology all kind of follow that same exponential phase. I'm not saying that it's going to be 12 months or 6 months, but if we're using platforms that have already been developed and are shown to be safe, they're going right into almost phase 3 trials with these. So the vast majority of the research that would normally be done at the basic level and setting up to get to those phase 3 trials is all being skipped, right? And so we're still going to have...
You have to do a phase 1. Even in accelerated, you have to do a phase 1.
Yeah, but it's a combined, right? Isn't it?
No, it's phase 2, 3. That's what...
Oh, phase 2, 3.
Yeah, but phase 1, you have to do it, because you can't go into 200-300 people immediately, because if you have side effects, you're screwed.
Yeah.
And then no one will ever take the vaccine. No, they're all phase 1. The mRNA vaccine is in a phase 1. The Chadox, that's great. Chadox, the chimpanzee adeno from Oxford, that's in phase 1. All phase 1. But then if it looks good, you could do a combined 2-3, which would mean dosing and large numbers for efficacy.
Yeah, I think the furthest I've seen is people trying to do some paperwork to be ready for that phase 2 as soon as their phase 1 is finished.
And how long do those phase 1s usually take?
Like 2 months, right?
2 months.
To just do the trial, not to recruit and do the paperwork and get approved, and then afterwards look at the data.
Analysis.
2 months,
right.
You want to look at them for 2 months. And now the virus levels are going down. You hear China doesn't have enough circulation to test its vaccines now.
Yeah.
That's so interesting. Yeah.
Will the U.S. let them test theirs here? What an interesting idea.
I don't know. No, I mean, politically, no, and that's never going to happen, but it would be good for public health. Yeah. And, Cindy's right. I mean, there's a lot of technology that has sped up, particularly the beginning part of the process. I mean, an mRNA vaccine, you just need the sequence. But there's no mRNA vaccines licensed, correct?
Correct.
So we actually don't really know the safety profile in humans.
That's right.
No.
Right.
So,
and that's why they're working on all of those right now.
So anyways, just a little, blip about.
What is the longest vaccine what's the longest time a vaccine ever took, Steph?
Well, I'm gonna say, based on your background, the polio vaccine.
50 years.
50 years. Now,
from 1906, 1908 to 1957. And lots of false starts. Now, granted, we are better in technology, as you said, right now, so it shouldn't be 50 years, but we still don't have an HIV vaccine.
Yeah.
Yeah.
And it's not for lack of trying, right?
That's
no,
right.
definitely not.
Yeah. So we'll keep people updated. Each week we can do a little vaccine brief, even if we start to diverge to different topics, because it will be, it's obviously something on people's minds, but that, Vincent, is interesting that they don't have enough circulation in China to be able to test that vaccine. That's interesting.
Yeah, now it's becoming a political issue, right?
Yeah,
because they want to go somewhere, and this country is destroying it. What do you call that when you destroy your ties with a country? And
diplomatic. Well, if you think of the term, you can let us know.
So that's unfortunate, because I have to say that when Albert Sabin made his polio vaccine candidate, the U.S. wouldn't test it. They said, no, we have IPV. We don't need it. So he went to Russia. He went to Chumakov's parents in Russia who had visited because they were interested in infectious vaccines. And they said, sure, come over. And they did 100 million plus people clinical trial in Russia. Sabin brought over his viruses and they did it. Can you imagine?
Interesting.
I mean, that's because they had the people and the wherewithal to do it. Why can't we do it today?
Yeah, that would be great.
People have to fight. Burning your bridges behind you. Something like that. And then you have to turn around and go back and your bridge is gone.
Yeah. Yeah. For public health, it definitely is not ideal. So we will have to keep an eye on that. And that leads to, immunity in general and vaccine, and are you immune? And Cindy might want to talk about this because she has recently taught it in her class, but there's been a lot of conversation about serological studies. We have talked about serological tests, and in between, I think our last episode and this one, there was a couple serological studies. I think they're preprints. I don't think they're published, but their preprints came out of Santa Clara and then Los Angeles. And there was a lot of conversation about whether or not those accurately predict seropositivity. And can you do that with these tests? Because there is, a sensitivity issue.
Right. So I think that there's a couple of really important points related to this. Well, the first one is that it seems from a lot of these studies that there is prolonged detection by PCR, right? So we've already discussed, at least in feces, whether that would indicate that you're transmitting or not, because people aren't doing the plaque assays, right? But the idea that, we seem to have detectable virus for significantly longer with this particular infection than with some other infections. And more importantly, is that some of these studies are showing that even if people seroconvert, that means their immune system has now recognized the infection, starts to make antibodies, we can still detect the virus bits that we were talking about before for quite a long time. And so the other issue is that it seems like some people just aren't seroconverting. So even if they are infected, their body is having a hard time recognizing this infection and inducing an immune response. So those are two issues. But then the important issue that everybody wants to know is, once we seroconvert and you become PCR negative, so this means your immune system recognized the infection and presumably you have now cleared the infection. Are you actually immune, right? This is the basis of vaccines. This is the basis of what we talk about with memory in the immune system. But the answer is we don't necessarily know yet. So there's many different reasons why. With flu, we know we need the new vaccine every year, but that seems to be because the virus itself changes, so our immunity doesn't protect us, right? But what if our immune response to this virus doesn't fully protect us? Now, that gets into some nuance, right? Because even if you're quote unquote fully protected, we never fail to be infected again, right? So if you're going to breathe in more virus, it will go into a couple of cells likely, but you're not going to transmit much and you won't get sick. But the question is, what does the seropositivity actually tell us? And can we use this to give these people these, passports, right? That's the whole idea, that they want to use this. They want to say, if you're antibody positive, we now know you can't be infected, you can't infect anybody else, and you're good to go. You get a passport, you go out in public, and you don't have to worry about things anymore. And I think there's a lot wrapped up into that we don't really know yet.
Yeah. Wow. That's a great explanation. And the immunity passport thing, because we don't know protective levels. We don't know if that associates with neutralization of the virus and just how different people are in terms of their immune response in general. I am going to read an email that was sent in because it has to do with this immunity passport conversation. Then we can kind of go around and give our opinions on that. But so this email is from Jesse Mangold. Side note, he's going to his MD PhD, but he's a undergraduate at Duke who works in our lab. And he said, "Dear hosts of Immune, thank you for continuing to deliver timely and trustworthy science communication that I can rely on to cut through the noise. My name is Jesse Mangold, and I'm a research tech at Duke Human Vaccine Institute. And he's an incoming first-year MD PhD at Mount Sinai. So I think he'll be potentially involved in some of the exciting work that's going on in Mount Sinai in regards to viral immunology. What I miss the most about the lab while working from home is listening to Immune while working in the hood and having the luxury to pick Steph's brain when I have a burning question. And I have to say, walking into the lab and hearing your own voice is a bit disconcerting. It takes me a minute to realize that it's me. And then I typically will just say, you know what, Jesse, you keep listening. I'm going to go over here because it's just, listening to your
It's weird.
It's very weird. Makes me sweat. Anyways, this time I will write in, Big Permar and Fuda, and those are our PIs. Lab style shout out to Steph. Thanks, Jesse. This time I will write in with a question. What are the key do's and don'ts around the design of a SARS-CoV-2 population serosurvey and their interpretation to inform policy? Do you have concerns with the circulating concept of immunity passport to return to worker society? For example, a biased sampling method could reinforce existing structural inequities by conferring more immunity passports to those people with greater access to testing. This brings a whole new meaning to the term immune privilege. Looking forward to hearing your insights. Thanks and stay well. Yeah, that is a huge part of it, is access to testing and those populations that are not going to have, they generally don't have as great of access to healthcare. So then you're determining their ability to work based on that. In addition to what we described, we don't even know. And for most infections, I don't think we would ever know enough to make this a public policy.
No, I agree. And I also really worry when I hear about this discussion of immunity passports, not just about thinking about people with greater access to testing, but I wonder about people who are seronegative, who really want to go back to work and who now seem to have some sort of incentive to go get themselves infected.
Right.
Like a party.
That's right.
Yeah, that seems like a really bad idea to me. I really want to go back to my lab, and I do not want to be in a situation where I have to think, should I go do something dangerous to get infected?
Right.
That just seems like a terrible idea.
It is a terrible idea. But also, there's just think about who is in the forefront of interaction with the public. These are our people who are working in grocery stores and restaurants and things that are still open right now. And so if they're negative, does that mean that they're not allowed to work anymore? These people are not being paid a lot anyway, and they have been forced to work through this whole thing. And now if you're going to tell them only if they're positive they can work, I just think that there's going to be a huge societal inequity if something like this were put in place. And I would say, like, one other issue that I think is really important to consider when we're talking about these kinds of serological testing, or either any testing in general, was, as I was talking about this with my class the other day, there's a really important thing that we often don't think about, and that is the prevalence of the disease actually in the population.
Yeah.
Right. So if you have a high prevalence, so there's a lot of people that are actually positive, when you test, there's a good chance that a positive test is actually positive. And there's a reasonable chance that a negative test is actually a false negative. But on the flip side, if we only have a small portion of the population that's actually infected, so the prevalence is really low, if you have tests that aren't so specific, now you all of a sudden have people testing positive that aren't positive.
Right.
Right. So, but a negative is a good chance that it's a negative. So as we're testing the population now with relatively low prevalence in a lot of areas, then a negative test is probably reliable. But as we cross a threshold and a lot more people are actually positive, a negative test becomes less reliable.
Yeah, for sure. And, I don't know if these conversations were brought up because I don't know who started these conversations of immunity passport, if it was actually from a government official or if this was just general think pieces that people were publishing. But people have seemed to come off it. And I think it's because what we're seeing from these sero surveys is it's very difficult, one, to get a test with the specificity that you want, and then two, to rely on that. So I don't know if we'll have these conversations much more, but
I don't know. I think those conversations are still happening. They're happening locally here in Ithaca because, and at Cornell University and other universities, because they're trying to figure out a way to reopen, right?
Yeah, that's true.
So
and healthcare workers.
You need something.
Right.
Well, we do have a
something's better than nothing.
We do have a test called PCR that will tell you if you're infected.
That's
and that
right.
can be used to, quarantine people and do contact tracing and limit outbreaks. But we don't seem to want to do that in this country.
Well,
and I don't get it.
Yeah. I mean, if we can't even implement widespread testing to see if anyone even has the virus, then to tie it to our ability to work, we wouldn't have the capacity.
These seros. Go ahead. The serological surveys historically have never been used to determine who is susceptible or not. They've been used to say who's been infected or who's encountered the virus, right?
Right.
And now for some reason we want to use them to say you're okay. I don't get this. But I don't get where this is coming from. It's the wrong use of a sero survey.
Yeah, I think people really are just wanting to, go back to work and reopen the economy and make money. And I can appreciate that, but it would be irresponsible too.
Yeah, I think that it's in some ways sort of wishful thinking that people want to get back to work and open things back up. But I just don't have any idea of exactly, how much antibody is needed to be protected and whether or not we're measuring protective antibody versus just any old antibody and things like that.
Yeah. Never for any virus we able to do unless you do a neutralization assay. Right?
Nope. Yep.
So on Twiv, someone wrote in from, I don't know, Taiwan and South Korea, and they have never shut down. They've done extensive PCR
that
testing.
Was Taiwan.
Yeah, and the other day Jeff Shaman talked about the South Korean experience where using extensive PCR, they could limit infection by quarantine and contact tracing.
Yep. And
And it seems to me we're just forgetting about that, and we shouldn't.
Nope.
Yeah. So I think that's a good point. We could transition. We could read a couple more email, or we could kind of roll into this last section of, we have a couple more sections, but testing, prevention, and control. Do we have any preference? Whether we want an email?
Let's do a couple of emails.
Sure.
What do you think?
That bounces us back to, last one was Matt, so that bounces us to Benjamin.
Right.
Vincent, if you can read it if you want.
Benjamin, good afternoon. It's 62F in Washington, D.C. I work in a lab for the Department of Defense, and while I am not working in immunology or virology, your podcasts have encouraged me to look into it.
Do it.
You have been talking on both Twiv and Immune a lot about the possible lack of sterilizing immunity that could be acquired from infection, from immune response to SARS-CoV-2. What impact would this have on the efficacy of a vaccine? Do vaccines still work, at least through the mechanism of artificially increasing herd immunity, if they don't give sterilizing immunity? So maybe we should stop and answer that.
Yeah, I think so. I mean, if you're going to decrease overall, because we could assume that this vaccine will not cause sterilizing immunity, and I don't know that most vaccines do. I still think you have low circulation. So, but if you're lowering it, then you have less people infected, and then you can reach whatever your herd immunity percentage is. I still think you can reach herd immunity with a non-sterilizing vaccine.
Yeah, I think if you look at the experience with the common cold coronaviruses, and Jeff Shaman just put out a paper on this. They've been doing a survey in New York City for infection with these viruses, and they find people get infected. They get low level of serum antibodies, and then they get reinfected every year with mild or no disease. So that a vaccine could do the same thing. It could just give you this low level immunity. You don't get sick, but you get infected. The virus circulates. I don't think that's a bad situation, right?
And I should clarify, just in case, because I'm assuming people know what sterilizing immunity is, but it's the idea that you would not get infected. There would be no replication. You would have no symptoms. And then non-sterilizing immunity is you maybe would get low level of infection, but you're potentially not spreading it. It would be mild in general.
Yeah, I would agree with that. I think that, this is something that I encounter. I don't know about you, Vincent, with the anti-vaccine crowd, is that they seem to think that vaccines are useless and herd immunity doesn't exist if people could potentially still get infected, even if they're immunized. And I think that it's a really hard thing to explain that, if you have a more robust and a more rapid immune response because you have pre-existing memory, you're going to dramatically reduce the amount that you would spread. Mm-hmm. And so you decrease the load in which you would be able to transmit. So it still works. But,
the problem is,
it's an argument.
I think people equate infection with getting sick.
Yes.
In a way. And they don't understand that you can be infected and not get sick.
That's right.
And in fact, we have an email by someone says, how can that be?
Yes, right? I would say that is one of those things that's a little bit of a soapbox that I think all of my students get to hear a lot when I'm teaching. I talk about it all the time, is how infection does not equal disease.
Because, that, as you well know, Cindy, and the point of a vaccine is you get an immune response without getting sick. Right. And then you subsequently get infected. You may get some virus in you, but you don't get sick. So why don't they get that? I don't understand. Well, they don't want to get it, really.
They want to say that means that if you don't get sick, that you will still continue to spread, and so that the idea of herd immunity is irrelevant. And I don't, and that's not true, because if you have someone who doesn't have pre-existing immunity who would shed 100 particles, and someone who does have pre-existing immunity would shed 1 particle, the probability of transmitting to another person is dramatically reduced. So that's exactly what herd immunity is. While we're talking about this, one of the other arguments is that if you are targeting pathology in an individual and not necessarily transmission. So let's say we have some vaccines that are generating immunity against the toxin rather than the bug itself. And they're like, oh, but that's only going to affect the toxin and not the bug. And how do you explain to someone that, but the toxin is there because the bug uses it to gain a foothold on the immune system and the host. And so if you block that, the bug is crippled and doesn't replicate as efficiently and doesn't transmit as efficiently.
Yeah, it seems like what I've learned from science communication is it's all about the long game, but that's very hard to build these conversations because they're so nuanced. I mean, gosh, I feel like the more I learn, the less I know, kind of thing. So to be able to translate that to the public it's very challenging. But I guess persistence, right? And having over 600 episodes of podcasts helps.
Helps.
Yes.
Did you want to read the second half of that email?
Yeah, then Ben continues. I'm wondering if you knew of anyone doing cool vaccinology, immunology, virology type work in a fish or non-mammalian animal system that you could suggest, and looking into grad schools, and any suggestions would be appreciated.
Steph?
Yeah. Oh, yeah. So if you listen to Immune 28, we had an excellent time interviewing Irene Salinas, and she is a really good immunologist working in fish models and non-mammalian animal systems at University of New Mexico. So you should contact her, listen to that episode, and see what if there's any opportunities there. And I visited that campus. It's beautiful. It's in Albuquerque, and she has fish in her office. There's fish downstairs. There's fish all over the place. It's so cool.
We also at Cornell have some researchers who are doing fish research as well, which is kind of cool. So, we're at a vet school. So I would encourage you to look at some of the vet schools, the veterinary schools, because they would have companion animal or non-traditional model systems or, systems. There's also people who use zebrafish for various different innate immune studies. And then Brianne, you were mentioning somebody that I know also who's really fascinating.
Yeah. So one of my friends who did a PhD in virology then wanted to look at fish immunity and did a postdoc at Maryland with Martin Fladnick. So
he does fantastic really comparative genetics work. Yeah.
Yep. And so I know she was really enjoying his lab. And actually one of my students in my research lab got into the Cornell program and was particularly excited about all of the opportunities. That was the thing he liked about Cornell, was all of the people who were doing sort of non-mammalian types of immunology.
And the cool thing is like they're doing it in the natural animal, right? So we often think as immunology, we look for animal models, but they're actually studying the disease in the animal for the sake of understanding and developing therapies for the animal, which is different than I grew up as an immunologist, but it's really fun to see. And sometimes there are things that they're trying to develop in the animals that they can translate into human, because there are certain studies and samples and things that you can do in animals that you can't do in human patients.
Yeah, for sure. Cool. All right. Well, we could do Lizzie is next.
Sure. Yeah, go ahead.
Go ahead. Okay. Hello, immune team. This is quite long, so we could answer questions as we work through this. I'm a healthcare worker currently assigned to care for patients in an ICU designated solely for COVID-19 patients. I work in one of the academic medical centers enrolling frontline healthcare workers in a randomized hydroxychloroquine versus placebo study through the HERO trial. I and many of my coworkers are intrigued by the study as a possible opportunity to both get tested for SARS-CoV-2 to see if we are pre-symptomatic, and potentially, though not proven, to prevent becoming infected or ill, which is a daily concern for us. While I appreciate reading about the possible side effects of hydroxychloroquine, including GI upset and QT prolongation arrhythmias, I wanted to do a little more digging on my own before I decided to enroll. I found many articles about the use of hydroxychloroquine to treat an array of autoimmune diseases and other non-malaria disease processes. I'm wondering if you all would be willing to discuss the immunomodulatory effects of taking hydroxychloroquine for a person who is healthy and likely not infected with SARS-CoV-2. I gather from my research that there's quite a bit that happens at the cellular level as the body responds to this drug. I'm curious about the potential silent side effects. So we can tackle this, and then I could read the rest of her email in a bit.
I can say a little bit.
Yeah.
So first of all, thank you, Lizzie.
Yes. Thanks, Lizzie.
Thanks to you and all of your coworkers. We can't say thank you enough. Yeah. I have studied innate immune receptors and signaling and regulation mechanisms for a long time. And so one of my interests is in the innate sensors that are recognizing nucleic acids, and Brianne, you do too. And so some of these are toll-like receptors, which we've talked about in the past. And it's interesting because they seem to recognize their nucleic acid ligands inside endosomes or phagosomes. And hydroxychloroquine does block signaling through those receptors, and it's thought to have something to do with pH changes, neutralizing pH and so forth in the endoplasmic, in the endosomes and the phagosomes. And so that has been studied as one of the potential mechanisms why those drugs are good for autoimmune disease. because we know from studies from Anne Marshak-Rothstein and others that TLRs, especially these nucleic acid sensing ones, when they recognize their ligand in B cells, they prime the production of autoantibodies. And so there's this feed-forward loop where nucleic acid recognizing antibodies bind nucleic acids, internalize them, trigger the TLR, and promote more production of autoantibodies. And so the drugs work in part by disrupting those signaling processes. And so that's one of the reasons why they really work well for autoimmune diseases. They're also naturally, quote unquote, immunosuppressant, whatever that might mean, whether that's actually through their TLR mechanism or through other mechanisms. I'm not sure. So I don't know what other people want to mention on that too.
I mean, I think the only thing that I would say is my understanding is that the autoimmune diseases that are used that this is used against are predominantly things like lupus.
Yes.
And lupus might have a little bit of a nucleic acid part to its pathology. And so it may not be something it may be working specifically for lupus because lupus may involve this nucleic acid TLR endosomal signing as part of its pathology.
That's right.
Yeah. And
what's going to happen to a healthy person? I don't know.
I don't know either. I think both of what you said is excellent, and I don't really have any additional things to add other than if it were me, and not that you should do what I would do, but I think that there are many upsides to this, in addition to seeing if you they say get tested for SARS-CoV-2, and would I be curious if she's going to get tested for the virus or if she's going to get tested serology for antibodies. That would be
I would guess both.
Probably both.
Large trial.
The only reason I didn't know is because I remember Daniel Griffin, MD on TWIV, was talking about their hydroxychloroquine study, and they just went ahead and Vincent, correct me if I'm wrong, just did PCR testing because they hadn't picked a serology kit yet they were happy with for the PROMIS. I don't even remember what his trial was called, but either way, both would be great. And potential silent side effects,
patch trial, patch study.
Patch study.
Sorry.
No, that's fine. That's hard to say. I guess if they're silent, you don't have to, that's not something, it's something happening at the cellular level that it's not symptomatic. So, we're not MDs here, but if you think that it would be worthwhile and would be an interesting way just to contribute to science in general.
What do you guys think about this hydroxychloroquine?
Yeah.
I thought at first I said, that's interesting because if there is acidification required for spike protein activation and fusion, then that might block fusion, right? And on top of that, if immunopathology is induced in some small way through TLR signaling, which we don't know, that's totally hypothetical, then maybe it might dampen that. And, you would have dampened inflammation and potentially less immunopathology. However, a lot of the studies have come out have conflicting results. And so I'm glad that they're doing a larger controlled trial, but I'm a little concerned whether it's actually going to work or not.
So this patch trial, which stands for Prevention and Treatment of COVID-19 with Hydroxychloroquine, they're going to see if it prevents and treats COVID-19, right? So both see if it can block infection and if you give it to an already infected patient. And I think Daniel has said that the problem is it's immunosuppressive. Right. So if you give it too soon, you're going to get too high virus loads.
But
so you have to really give it later when the immune response is higher and the virus loads are low, but then it doesn't, if it's antiviral mainly.
Right.
Yeah. So that's why you need a really good trial. And the first trials that were published were not really well designed to address these nuances, basically. So his is really big, multi-state, and should answer these questions.
Right. And I think that it's important for us to look to our MD colleagues to understand some of the other things that are going on outside of the immune system. I seem to have read a bit about effects on the cardiovascular system with the heart with this drug. And that is really not my specialty. So I can't tell you how that is happening. But I think that that's something that we need to think carefully about in terms of use of these drugs in healthy patients and look to our MD colleagues to help us out a bit.
I would agree with that.
Yeah. So hopefully, what we were able to say maybe guides you in a way, but obviously there's going to be, consultation with enrolling in this. And so you would just want to make sure that you are already baseline healthy and don't have heart problems or already immunosuppressed in some way. So, because those are things to consider. I can read the second part. So on a side note, I'm a huge fan of TWIV and Immune, as well as the other podcasts, and have been interested in virology since listening to Vincent's Virology Lecture series in 2012. I read Spillover by David Quammen in 2015, and his chapter on SARS-CoV-1 has made me fear coronavirus pandemic ever since. As a result, I ended up joining the Communicable Disease Response Unit at my hospital the next year. Wow, that's great. This is sort of a volunteer unit used for quick mobilization of regularly trained healthcare workers who received additional training for potential Ebola or MERS outbreaks. I owe my personal preparedness for the current crisis we're facing to the Microbe TV team and can't thank you enough. I share your podcast with everyone on my unit, and I don't know what I would have done had I not been able to turn to your podcast for trusted information. Best wishes, Lizzie. Well, Vincent, that is so cool. That is very
great.
That is great.
Makes you keep going. Right?
That's right.
Yeah.
Wow. Thanks.
When you have all the naysayers and anti-vaxxers and people who don't believe what you say and think it's made in a lab, then people like this come out and you feel like,
I know the warm and fuzzies. Well, we should print this off. This little part.
Yeah.
If we're feeling,
I also think it was really nice that they had this kind of preparedness team that, it just goes to show you that a little bit of preparedness goes a long way, right?
Yeah, for sure.
Well, thank you, Lizzie, but also thank you to Lizzie's hospital for having us.
Thank you.
So we've been going for about an hour and 15. Do we want to just wrap up with emails and then have a part 4? What do we think?
Yeah, sure. Let's do that.
Sure.
Okay.
Maybe Brianna's next, right?
Okay. Jacob writes, I have enjoyed both your virology and immune podcasts. They have helped me push my understanding of both subjects to a much higher level. I recently read a paper on how SARS-CoV-2 infects T cells. I came across this paper by accident while I was searching on another subject. The paper is enclosed. Is this a reasonable explanation of why we are seeing lymphopenia in COVID-19 patients? I have never heard of the CD147 receptor. Do you know what role it plays normally in T cell function? I have been following this story since January. I find it very interesting. It gets more interesting every week. In your most recent immune podcast, a question was asked on the role of proning. Patients are turned on their stomach every 4 hours to help match blood flow to open alveoli. This decreases shunting, which is a major cause of hypoxemia. When lying flat, alveoli on the front of the chest are open, but there is less blood because it has pooled in the back. So you turn the patient over so blood flows to where the alveoli are open. Then you switch them back. I'm interested in your thoughts on the paper. Thanks, from Jay. So I'm looking at the paper now.
Well, if I remember correctly, because there's been one big T cell paper that kind of popped out, and I think that it was roundly criticized by immunologists.
This is that paper.
Okay. So They really did not show that infected primary T cells. Maybe, I don't remember, maybe they showed a T cell line.
They do. They use a T cell line and show infection. And there are some questions about some of the ways that they show infection.
Yeah.
And I think these are not productively infected. They don't produce new virus particles either, right?
I don't think we have evidence that yet it infects lymphocytes.
So
I still got interested because I was like, CD147, what is that?
Yeah, this is the one that I sort of ranted a little bit about one of their figures being not the way I would have done my flow cytometry.
So what's the connection with CD147? Do you know?
Well, they're saying that it's a receptor, but all I know is I did a little bit of digging, and it's expressed on a couple of different types of T cells, but primarily on T regulatory cells. And so it's thought that it might have some role in regulation of T cell responses.
And then mostly preprints.
They say we recently reported to be a novel invasive route for SARS-CoV-2, CD147, which as far as I know, it's a preprint nobody is buying.
No.
Well, that paper that he put up is in Cellular and Molecular Immunology.
Published, yeah?
Yeah. In a Nature journal.
No, I'm talking about the CD147 as an alternate receptor.
Right. Yeah. So they cite a preprint in this cellular molecular immunology paper.
Oh, okay.
Citation number 9.
Ah, okay.
Yeah, and so I don't think that CD147 has been shown to
I agree.
play a role. We'll have to see. It's very hard.
But it could still infect T cells, yeah?
Yeah, I mean, sure. Maybe. Yeah, I think you have to have a high bar when trying to determine, receptor and actual productive replication of cells. So
right.
I don't know.
I asked Susan Weiss last week on TWIV if T cells are infected. She said no.
Oh, really?
Remember, Brianne?
I do remember, yes.
I mean, at least I don't think we have evidence. It's not to say it's impossible, but
no evidence.
I would agree. There's no evidence, and I don't really think that this is.
So is it the virus doesn't get in at all?
Well, according to this paper, it gets in, but the cells don't make new virus particles, right?
So my question would be: do you need to? Because if you make viral proteins, and since it's an enveloped virus, if any of those viral proteins get to the surface, you could still have depletion of T cells, even if they're not productively infected. Well, replicating and secreting virus, right?
Sure.
And so even if they just get some proteins,
the viral proteins could kill the cells themselves, right?
Right.
That too, yeah.
Sure.
So yes, it's possible, but these are cell lines, you guys said, right?
Yeah. Yes, it's cell lines, and I think we need some more evidence.
Yeah, I would agree with that.
All right.
Do we do one more?
Sure. You want to take that one?
Sure. So Marian writes, Dear Immune at Microbe TV and the team over there, I hope this email finds you well. I have been following your organization's work and want to let more in Asia learn from you. If you have more recent findings, do let me know. I want to disseminate only the best science and medicine in these times. So he goes on to provide some resources that he has available to him. And in partnership with the World Health Organization's Global Outbreak Alert and Response Organization, GORN.
I thought that when I read that quickly, I thought that said groan. And I was like, not a good acronym, but okay, got it. GORN.
My medical school has launched 2 public education initiatives on COVID-19. We have a webinar series that reaches out to doctors and scientists and shares the most up-to-date information from medical and research experts on the ground in Singapore. So very real and transparent. And then also there is, where was the second part of that? There's educational comic strips for lay public communication. And there's some links here that I suppose we could put up to their information that they are putting out for their webinars.
Mm-hmm.
Well, I'm glad if he or she, I don't know if Marian's he or she.
Oh, that might be a woman.
It might be. Yeah, no, it's fine. Is looking towards microbe.tv for information. That's great. And hopefully can help facilitate education. So yeah, we could post these if you think that would be fine with me.
These are cool comics.
The comics are cool.
The COVID-19 Chronicles. So National University of Singapore, I visited last December.
Oh, nice.
It's a campus of Duke, Steph.
I was going to say it is. Yeah. Mm-hmm.
Duke NUS. Yep.
Yes.
That's where Linfa is actually, who we mentioned earlier.
Yeah, Linfa Wang. And then I don't know if you're familiar with Ashley St. John. She does.
Yeah. I met with her. She was on. I did a TWIV with her and a few others, and I still have to release it.
Oh,
because,
really? Okay. Well.
Yeah, she was really very interesting.
Yeah, for sure. I know it's hard to release non-COVID related.
I'm going to start doing it because otherwise they'll be 2 years old by the time they're out, and it's not good. Can we take a few more, or you want to stop?
Oh yeah,
let's go.
I don't know who's next. Steph,
I can do Steve. Yeah, sure. Hi, Vincent et al. Good to see these early results from China on infusing critical COVID patients N of 5. So, I mean, let's be a little careful with what we say of good results with convalescent plasma. This reminded me that I have been wanting to ask about the business of using antibodies from donor blood in general. I had wondered why antibodies from another person weren't treated as foreign by our own immune system so that we'd make antibodies to the antibodies or anti-antibodies. And we can answer that question first. So I think that is something that is considered as a potential risk factor, although you would be infusing convalescent plasma to do some sort of therapy. So the risk would be low, and the reward, potentially saving your life, would be high. So that's like a risk reward ratio. But also, I don't know if convalescent plasma would be in your blood long enough to develop autoantibodies. And I don't think there's much evidence for that being true. I think it's more theoretical. What do you guys think?
Yeah, I think the idea is that, yes, those antibodies would be treated as foreign by your own immune system, but you're probably not getting multiple doses of them and having them over a really long time. So it's entirely possible. You could imagine kind of theoretically if you got convalescent plasma, and then 5 years down the road, you got SARS-CoV-3 and you happen to get convalescent plasma from the same person.
Right.
Which, is a stretch. Perhaps you would have antibodies to their antibodies and make a response. And I talk about this because we do use passive antibody therapy, where we're getting antibodies from other organisms, in a few different cases in humans. One of them is black widow spider bites. And so I always tell my students that, if you get a black widow spider bite, and then you get one again, you might have trouble getting passive antibody treatment. But that seems unlikely.
Yeah.
Okay.
I think that, the interesting thing is each individual develops their own repertoire of antibodies, right? Because we have the randomness of the recombination that we've touched on in the past. And so, you could develop antibodies to another individual's antibodies. Probably it's not going to happen to a huge degree. You're more likely, what you were saying, Brianne, about like if you're using protective plasma that was developed in another animal, then you're definitely going to have an issue.
Right.
But the other thing is we have to remember that it's not infrequent that humans can actually develop antibodies to their own antibodies. So we get anti-idiotypic antibodies, and they are a marker for autoimmune disease, and they can propagate. The disease are very, they're common in lupus once you develop lupus. So it happens, the tolerance is broken. So it's not unheard of to have something like that, especially if you have a bolus that's introduced all at once.
Right. And maybe people who have an autoimmune disease would be ruled out from convalescent serum.
Maybe. Yeah.
If they're more predisposed. So moving on and following on these new results and on general testing in the wider population seeming to turn up quite high prevalence of symptom-free infection, might it be possible to make transfusions mix from batches of plasma from recent blood bank donors rather than relying on those who had been recently ill, whom blood banks would presumably turn down as a rule? And so I think we also mentioned on a previous episode that some of the criterion for donating blood or plasma being recently infected is that you would want to have high titers. You wouldn't want to give somebody lower titers because maybe it wouldn't be protective, or maybe it would actually induce pathology if you were to get infected, although we don't know much about that quite yet. So I don't think it would be probably something people would do to take blood bank donors, just because at least for this, you would want the titers to be high.
Yeah, because you're going to be just diluting anybody who might have titers with lots of people who don't. Yeah. Unless the prevalence is really high.
And I don't know if we can say that there's a quite high prevalence of symptom-free infection. I still think
we don't know that yet.
Yeah, I think we know based on epidemiological evidence and other evidence that there is asymptomatic people, but I don't think we can say what the prevalence is of transmission from those people yet.
No.
Right.
Also, is it possible for people to be infected with the virus and neither be ill nor mount an immune reaction? Virus just goes through without gaining entrance into any cells. This way, some people could be immune to viral exposure without any evidence of this in their blood. And so, yes, I think that would be considered sterilizing immunity, what we discussed earlier. But I don't, in terms of a primary infection in a seronegative or at least a non-protected individual, I had always thought that was very rare, that if you were going to be infected, you were going to be symptomatic.
Well, I think it depends on the virus. There are certainly many viruses where you can be infected and have no symptoms. There are some viruses that 90+% of the human population seem to be infected with, and we have no symptoms. We often might mount an immune response, although I don't know that I have looked enough to say that we would always mount an immune response. So I think it is very virus dependent.
Oh, I see what you're saying. Yeah, you're right. I was thinking in the context of the bugs we know that make us sick, and would somebody, could they be infected with influenza and never have a symptom? But you're right. Like EBV,
well, in this case, they're saying not without gaining entrance to any cell. So I guess the definition of infection is that that a pathogen gains entrance to a cell and replicates.
Right. I usually think of infection as colonization.
Yeah, there might be some that don't go inside a cell, but yes, colonization is a better word for it. But I don't know though. Do any people have polymorphisms in the receptor that protect them?
Yeah,
wow, that's a good question.
Good question.
I haven't seen that yet.
People are looking at that.
Yes,
I'm sure.
Yeah.
I know there are a lot of polymorphisms in the innate genes that can protect against coronaviruses in general. A lot of the interferon-induced genes are highly polymorphic, but ACE2 specifically, I think we'll learn a little bit more.
And so I guess that would make sense. There are people who have mutations that make it so that HIV cannot, or certain strains of HIV are not able to enter their cells. And so in that case, they would be neither ill nor mount an immune response, as
that's right.
Steve mentions here.
Sure. And he ends this email. Hope these aren't too dumb questions for you and the I team. Definitely not too dumb. No questions are. Wash your hands and scrub under your nails too. Steve.
Cool.
You want to stop there? Is that good?
Sure.
Yeah, that's good.
Covered a lot of ground.
We sure did.
Okay.
God, there's a lot of emails.
There are. Brianne, this just might mean you just might be with us for a while if we continue our COVID.
That's fine.
I don't think.
But she definitely has to be here when we talk about the bat thing.
Yes, I know.
The bat stuff I think we talked about that paper on TWIV.
Probably. Yeah.
Really? So we'll try and get the bat guy.
But it's so cool.
Yeah. Vincent, do you want to reach out to him? You said he's at Fort Collins.
Yeah.
Okay.
Yep. I will do that.
Sweet.
So we should at least do one more where we go through the rest of the topics and some more.
Sure.
Yeah, part 4.
And then COVID pandemic will be over and we can go back to normal.
Yes, the COVID pandemic.
What are you going to do when you're not doing 4 TWIVs a week on COVID?
It's fine. I'm doing it to help people. Right?
Oh, I know.
I will go back to 1 a week. That's fine. But I think it's going to be a while because, well, maybe, if you look at the curves, they're going down and the models predict end of June is really negligible circulation. And so it might be quiet over the summer. We'll see. Unless when
in New York or everywhere.
Well, in New York, in many places, there haven't been many infections at all. Rural places, for example, like Ithaca,
Yeah, I was just hearing, so I don't know if we're still recording. You're going to put a break in here. But my husband is on local government official calls, and they were talking about we've had 1 case in the last 7 days.
Huh.
Yeah.
So, they're ramping up. We have thousands of tests being done and like 133 cases so far. Okay. So it's not.
You should monitor. That's a good thing to do. Monitor and make sure. And then you see a case, you quarantine them. That's the way to keep it down.
Yep. And they're doing a lot of contact tracing.
Good. That's what they should do.
Yep.
Everywhere, not just in Ithaca, but it's out of my control. I'm not the governor. If I were the governor, I'd be doing it more. I'm surprised he's not, frankly. But,
I think they're counting on the local health departments to be doing this.
Not sure that's good, but anyway. All right, Immune 31. All the notes and letters, microbe.tv/immune. Send us your questions and comments, immune@microbe.tv. And if you want to support us financially, microbe.tv/contribute. Today on Immune, Cindy Leifer is at Cornell University. Cindy Leifer on Twitter. Thanks. Wait, let me say that again. Thanks, Cindy.
Thank you.
Steph Langle's at Duke University. Stephanie Langle on Twitter. Thanks, Steph.
Yeah, thank you. This is fun.
And Brianne Barker's over at Drew University. BioProf Barker. Thank you, Brianne.
Thanks. It was great to be with you guys.
I'm Vincent Racaniello. You can find me at virology.blog. Music on Immune is by Steve Neal. Thanks for listening to Immune, the podcast that's infectious. We'll be back next month.